Analysis of Carbon Dioxide by Lauren and Dan. The respiratory system is responsible for the intake of oxygen to the blood and the removal of carbon dioxide. The cells inside the lung excrete a surfactant that reduces the surface tension of the liquid layer on the alveoli, which helps the lungs from collapsing on every breath. As seen in the fetal pig picture on the left, the lungs have not been inflated, so they are smaller and solid in comparison to the adult lungs. Breathing is also controlled by the medulla of the brain. Blood works with the respiratory system to maintain a balanced pH by using a carbonic acid bicarbonate buffer system. A buffer is a substance that resists pH change. Therefore, this bicarbonate buffering system holds the pH of blood at certain values, usually between 7 and 7.8. This is crucial because enzymes in the body system are sensitive to pH levels. Through the capillary network, substances can be transported through the body system. The picture here demonstrates the center of the circulatory system, which is the heart. The heart works by receiving oxygenated blood from the lungs in the left atrium. The blood then continues to the left ventricle, then the aorta, and out into the body. The right atrium receives the blood with little oxygen from the vena cava. The blood then travels to the right ventricle, then the pulmonary artery, before finally traveling back to the lungs where it is reoxygenated before repeating the cycle. In lab, the respiration experiment demonstrated the difference between carbon dioxide levels in the air and in our breath. When we bubbled air into a cup of lime water, the water turned cloudy, whereas when room air was bubbled using the pipette pump, no change was observed. Next, we tested the effect of exercise on carbon dioxide production. The same method of bubbling air was used. However, instead of lime water, phenolphthalein was used. Phenolphthalein changes from pink to clear under acidic conditions, so when carbon dioxide is bubbled through it, it produces carbonic acid, thus causing a color change from pink to clear. Note the procedural change that instead of adding phenolphthalein concentrate to each beaker, a preset base solution was provided. Here you can observe the color change from pink to clear, and on the right is the micropipette pump that was used to bubble room air through the solution.
Following the carbon dioxide experiment, we received the following data in a graph. The sources of each air are color-coded, so the light blue is exercise air, the normal blue is exhaled air, and the darkest blue is room air. It seems that the room air distribution seems a lot lower in general than the other two categories. But as for the difference between exhaled and exercise air, there is no significant difference. So there was no availability of a linear regression analysis. So we performed an ANOVA with the following results. Our null hypothesis was that the amount of carbon dioxide does not vary with its source. However, the p-value refuted this null hypothesis with a value of 1.7 times 10 to the negative 12, which gives us a near 100% confidence that the variance in carbon dioxide content depends on the source. So the way we measured how much carbon dioxide was in the solution, essentially the higher the number of drops you have to put in to return the solution to a base, the more acidic the solution was in the first place. And the more acidic, the more carbon dioxide content. Our hypothesis was that in increasing carbon dioxide content, it would go room air, exhaled air, and exercise air. The reason for this is that the room air contains a very low amount of carbon dioxide relative to what one person would directly put into the system. In addition, exercise promotes an increase in heart rate and oxygen exchange in the alveoli. So there's a higher concentration of carbon dioxide, and in general, your breathing rate increases. So the originally pink solution turned clear more quickly. The equation of this reaction is shown below. The theory is that an increase in the pressure of carbon dioxide will cause a decrease in pH and therefore make the solution more acidic. We measure vital capacity with a spirometer, which is shown here. The spirometer is essentially used by blowing into the tube, and it measures how far the inside bucket, which is surrounded by water, rises. And the increments up top that the chain is lined up with indicates how much volume or vital capacity is. Each about inch increment represents 1 liter. This is our distribution of vital capacities across the board for the entire class. There is a noticeable trend. However, there is a difference in frequencies. We performed standard deviation analysis, which allowed us to calculate a mean and therefore what the normal distribution was. And as you can see, there's a very rough bell curve in this histogram. We calculated the standard deviation to be about 0.8 with a mean of about 3.45. Specifically for the team, we were 2.3 and 3 liters. 2.3 liters was 2 standard deviations from the mean, which makes it outside the normal distribution. However, the 3 liter vital capacity is within 1 standard deviation of the mean, which makes it within the normal range. But both data points are below the average vital capacity for the class. Here's our list of references, mostly for photos, and the information about the respiratory and circulatory systems is retrieved from the document provided in Blackboard.